soluble resident ER proteins can escape from the ER. And what are two soluble, by resident protein, we mean a protein that lives in the ER that is responsible for modifications. What are two soluble ER resident proteins? Go ahead.
One of them we said was BIF.
One of them's BIF, right? And what's the other one? Or another one? Yeah. PDI, right. So BIP is, and then calnexin I sort of mentioned as a afterthought. But BIP and calnexin are chaperones. PDI is doing something different. It is forming those disulfide bonds. So again, all the proteins that are going to be on the outside world are heavily folded, heavily have disulfide bonds. And with some frequency, those are captured in COP2 coated vesicles. I don't know why this illustration has the COP2 coated vesicle as yellow. These are both supposed to be COP2 codes. And it's showing you here, these might be ER resonant proteins that are floating around doing their job of folding and whatnot. And with some frequency, along with the secretory proteins, which are shown in red, they're captured in these vesicles. There's nothing in the liquid that could really retain them back in the ER. They're captured up in COP2 coated vesicles. So these can, with some frequency, escape from the ER, and the cell has to have a way to return them. So how do ER resonant proteins remain in the ER? So what are ER resonant proteins? We've talked about two of them, BIP and PDI and calnexin. And Hugh Pelham, who was in the MRC, came up with a hypothesis for how these resonant proteins might remain in the ER. And his hypothesis was sort of based on Günter Blobel's lab. Remember that Günter Blobel is the one who came up with the ER signal sequence. And so his hypothesis sort of, because most science is done in incremental things and you're sort of inspired by what was done before, his hypothesis was maybe there's a signal, some kind of signal on ER resonant proteins that are responsible for targeting them back to the ER, for maybe going into these COP1 coated vesicles and targeting them back to the ER. It's a reasonable hypothesis, don't you think? Make it, and you can see how it comes from the global lab, right? So now what I want you to do is pretend you're in Hugh Pelham's lab, and he's run into the lab and says, I have this great hypothesis. You all nodded your head, so you think it's a good hypothesis too. You're the graduate students in the lab. What experiments would you do to test that hypothesis? And I want you to work in small groups and sort of just brainstorm. What experiments could you do? So go ahead. So what experiments could you all come up with? There's lots of different experiments. And assume you have an unlimited budget, lots of time, you're not bound by anything. What kinds of experiments would you do? Anyone? Yeah.
So my idea was that since the ER signal sequence we know is a hydrophobic patch, I would sequence both the proteins, I would sequence the proteins, and then I would
Which proteins?
For the BIP and the other resident protein.
Which are
PDI.
PDI and BIP. So you'd sequence those.
So I'd sequence those. I'd look for hydrophobic patches, and then create mutations in the hydrophobic patches.
Okay. So you'd sequence the resident proteins. Look for hydrophobic patches. Because you're very influenced by Günter Blobel's work that identified the ERSS.
Right.
Look for hydrophobic patches. And then do what?
And then just mutate them and
Mutate
see
them and
which
see where
ones
they
are
go.
important for binding to the code. That's cool. Put one method.
Okay, other experiments. Yeah,
I would do something similar to that, but I wouldn't necessarily focus on a hydrophobic patch because it's a soluble protein in the ER, and so I would we discussed that we would do like point mutations in proteins.
sequence, so you'd also sequence and then do point mutations, and what would you mutate?
So kind of randomly.
Randomly, okay. And then what would your test be?
The test is to basically to identify mutants that do not retain.
Okay. ID mutants that fail to return to the ER. And if they fail to return to the ER, where would they go? We haven't really covered this, but where do you think they might go? They end up being secreted to the outside world, so they're would just be a soluble proteins and are secreted. Okay, other experiments, yeah.
Right, so we were saying that we would sequence against all of the resident proteins, but this time compare for conserved sequences.
Okay, you'd sequence them,
Yeah.
proteins, compare the sequences,
Find conservative sequences.
and then what would you do?
Find conserved sequences, and then do knockout or mutations to those like, specific sequences and label them and see if they return those sequences that take
Wait,
away.
find the conserved sequences and then
Knock those out or
not
disrupt
change them, point mutations maybe, or whatever.
and
Point,
see if
I'm gonna
those-
just put mutate them,
if they do different things.
and then,
And then like label them and see if they return back or go secretion or.
uh-huh. Other experiments. Yeah,
I think we had a very similar pattern. We also like sequence proteins and then look for like conserved patterns. We expected that like it would be a unique pattern just like an ERSS. And if we based for that unique pattern, we could create an antibody immunofluorescence that we could monitor the movement via like ER to COP2 to COP1.
uh-huh, you'd create a fluorescent,
An antibody immunofluorescence
Uh-huh.
to attempt to watch the movement of this unique sequence
uh-huh,
and then see if that unique sequence is being exhibited in all three.
okay, so you would, you'd label the sequence that you found.
Yes.
Cool. Maybe not with immunofluorescence, because that would be, you need an antibody against the sequence, and then it would pick up the wild type, the regular one, as well as the mutant one. But what if we GFP tag and express the sequence? Okay, so I'm gonna put ID sequence, because it's similar to the other approach. GFP tag it, and ask where. Anybody else? These are great experiments, they're all doable. And this is really similar to what Alexis and Lexis found. And Alexis had the question, and that's why I sort of interrupted, when she said she was going to sequence line up the sequences. So get the gene sequence, line them up. So not sequence the proteins, but the genomes are already known. So line them up, look for conserved sequences, and then test whether that was. And before the testing, she wondered, is that an experiment to do sequence alignment? And it totally is. There are scientists who spend their whole time gazing at sequences on the computer. They never get their hands wet. And those are totally experiments. And then they might do the mutations, or they might give it to somebody else. So there's all kinds of experiments and all kinds of science that are possible for this. So these are really great experiments right here. This one right here, that's a wonderful hypothesis based on what the information that was known at the time looking for hydrophobic patches. But you might quickly find out that you'd reject this hypothesis that it was a hydrophobic patch unless that was the signal. But that would definitely be a graduate thesis, even ending right there. If you finished your 5 years and it was like, okay, the thesis is it is not the hydrophobic patches, but maybe if you had more time, you'd go on to do these other experiments. And we had 2 ways to sequence, to figure out the sequence. One was sequence alignment, looking at this, but we also had the experiment of doing random mutations. And that's actually, both of these experiments were done. So this one was done by comparing the sequences, but at the same time another lab, or maybe it was the same lab, I can't remember, used yeast to randomly mutagenize proteins and ask what was deficient in yeast cells that could not return ER back to the ER proteins back to the ER. And through that random mutagenesis, they not only were able to identify signals, but also the accessory proteins that were able to do that. And then this identifying sequences and doing a GFP tag, that probably would work. At the time, GFP wasn't available. So this is wonderful. You all came up with the actual experiments that were done, some of which were Nobel Prize winning work. So pat yourself on the back. Yeah. So let's look then. Let's look then at the actual. So we've covered 2 things here. One is in comparing the sequences, we found common sequences that are suggested. And then you all also came up with experiments that suggested that a sequence was necessary for proper sorting. So first you had a hypothesis that this common sequence would be, maybe you'd find hydrophobic patches on the protein. Undoubtedly you would. Remember, these are soluble, but they're going to have hydrophobic patches because that can be in the interior of any protein. And it could be a signal. And then you can test whether it's necessary by altering the signal and seeing where it goes. So let me show you the actual experiment from the original paper right here. What Hugh Pelham's lab did was he took BIP and PDI, as both of you were suggesting. The reason why, as many of you were suggesting, the reason why you might choose BIP and PDI is they're completely different proteins. BIP